So the topic for today is amino acids and proteins. There are 20 amino acids that exist in nature. The list of the 20 amino acids and the representation, alphabetical representation of those amino acids is given here. The list goes like glycine G, alanin A, valine V, leucine L, isoleucine I, proline P, methionine M, phenylalanine F, tyrosine Y, tryptophan W, lysine K, arginine R, histidine H, aspartic acid D, glutamic acid E, serine S, threonine T, cysteine C, asparagine N, and glutamin Q. Out of these 20 amino acids, the amino acid arginine and histidine are semi-essential amino acid, which means that they can be synthesized in children's body. However, they cannot be synthesized in adult's body. Selenocysteine is an amino acid which is present exclusively in bacteria. Now, if for example, in a question we are given uh, 2 or 3 or 4 amino acids and they ask us how many types of tetrapeptide can be formed using those amino acids. So we calculate using this formula in which we find out the factorial of the number. For example, we are given 4 amino acids over here. So we'll give the factorial of 4. 4 into 3 into 2 into 1. So we get 24 as an answer. That means using these 4 amino acids, we will be able to form 24 tetrapeptides. Again, case 2, let's say one kind of amino acid is same, it's in repetition, and the other 2 are not in repetition. So again, we have 4 amino acids, but in this case, since one is same, 2 are same, so we'll calculate using this formula, which is the total factorial divided by factorial of number of same amino acids. So total factorial will be 4. For factorial will be 4 into 3 into 2 into 1, and factorial of number of same amino acids, we have 2 same amino acids, so that will be divided by 2 into 1 and we'll get the answer. Same goes for any kind of aptitude kind of question, so we can use the same formula for calculating the number of combinations that can be formed. So in case you're given a palindromic sequence which goes same in both the direction, from front to back and from back to front, in that case, if you're asked how many types of palindromic sequences can be created, so you use the formula n raised to the power 2 or n squared, where n is the number of given amino acids. So in case we are asked that how many palindromic sequence can be created using 20 amino acids altogether, the answer for that will be 20 raised to the power 2, that means 400. So we can create 400 palindromic sequences using 20 amino acids. We have calculated that using the formula n raised to the power 2 or n squared. Coming on to the structures of amino acids, the non-polar aliphatic R groups, basically hydrophobic amino acids. The structure of those are given here. First one is glycine. Glycine represented by G, and the structure is this. It's the only amino acid which is optically inactive, and it's the simplest kind of amino acid. Optically inactive means that the carbon atom in glycine is not a chiral carbon atom. That means that it is not an asymmetric carbon atom, because it has 2 H groups. 2 similar groups are attached to the carbon atom present on glycine. The next structure is of that of alanine. The side chain, basically R, is CH3. In case of valine, this is the side chain CH3, CH3. In leucine, it's CH2, CH3, CH3, the side chain. Then isoleucine, again, it's an isomer of leucine. The position of carbons is changed. Proline is a ring structure which contains secondary amine. It's a cyclic amino acid, and it's also known as amino acid because the amine in this amino acid is secondary amine. Secondary amine is called imine, And if this proline is present in a polypeptide chain, it reduces the flexibility of that chain. So if we talk about antibodies, the hinge region in antibodies, they are rich in proline and cysteine. And all proteins जो है, they start with this is just a general fact that all proteins start with methionine, but they change after post-translational modification. And the most abundant antibody in human is IgG. Um, daily production of daily maximum production of antibody is IgA. And in IgM and IgE, hinge regions are absent. So basically, they might not contain proline or cysteine in the hinge region. This is the general information. Okay, the next structure which we have is of methionine. Methionine is mostly a start codon, which is same in all proteins, which the codon that codes for methionine is AUG. Methionine is a sulfur-containing amino acid. The structure is by normal CCO, NH3H, and then the side chain comprises of CH2, CH2S, and CH3. So ethylene, which is the only gaseous hormone, it is formed by methionine. Now, again, general information, pulses, they are deficient in the sulfur-containing amino acids, that is cysteine and methionine. And cereal grains are deficient in lysine and tryptophan. Moving on to non-polar aromatic R groups wale amino acids. So we have here phenylalanine represented by F. As we can see, we have a non-polar aromatic R group here, which is CH2 and attached to the benzene ring. And when this phenylalanine is acted upon by the enzyme phenylalanine hydroxylase, it converts into tyrosine, which again contains the non-polar aromatic R group. So, yeah, deficiency of phenylalanine hydroxylase, it leads to a disease called phenylketonuria. Phenylketonuria is an autosomal recessive disorder. Also, this is an example of pleiotropy. Pleiotropy basically means that problem in one gene will result in multiple effects. So, for example, if we have a deficiency of deficiency of enzyme phenylalanine hydroxylase, tyrosine will not be created. Phenylketonuria will happen, which is obviously autosomal recessive disorder. But in case tyrosine is not there, a lot of other multiple effects will be created because tyrosine is involved in creation of or production of many things in the body. So tyrosine is involved in thyroid gland hormone production, dopamine production, which is basically neurotransmitter. Epinephrine and adrenaline is formed by tyrosine, and the skin pigment melanin is also formed by tyrosine. So in case tyrosine is not formed by the deficiency of phenylalanine hydroxylase, all these things, thyroid gland function will be disrupted, dopamine will not be produced, and the dopamine deficiency leads to a disease called Parkinson's disease. Again, the epinephrine will not be formed, and also the melanin, which is the skin pigment, it will also not be formed in a proper way. We can get hypothyroidism or hyperthyroidism. Basically, the thyroid gland does not produce the required quantity of thyroid hormone. So hypothyroid is that thyroid gland is not making enough hormone, while in hyperthyroidism, a large amount of thyroid hormone is produced. In case of children, the hypothyroidism condition is called as cretinism. In case of adults, this condition is called myxedema. And even hypothyroidism, there's a autoimmune disorder which is called Hashimoto thyroiditis. In case of hyperthyroidism, it's called Graves' disease or exophthalmic goiter, which is also an autoimmune disorder. Next structure is that of tryptophan. It contains indole ring in its side chain, and auxins are produced from tryptophan. The structure of auxin is basically called IAA, which is an abbreviation for indole 3-acetic acid. Again, tryptophan is also involved in production of serotonin and melatonin. UGA, which is the stop codon in a lot of peptides, these codes for tryptophan in mitochondria. Again, Tryptophan and tyrosine and so to some extent phenylalanine, they absorb UV light, and these are the reason why proteins absorb maximum light or show a strong absorbance in at a wavelength of 280 nanometer. So basically when we study the absorbance, the wavelength, we know that proteins ka absorbance is taken at 280 nanometer. That is due to the aromatic amino acids, while the nucleic acids ka absorbance is taken at 260 nanometer, and it is due to the heterocyclic bases. Moving on to Beer-Lambert law, the instrument used is spectrophotometer. In this law, it basically states that absorbance is directly proportional to the concentration, and the absorbance is also directly proportional to the path length. So basically, absorbance is equal to epsilon multiplied by C multiplied by L. Epsilon stand for molar extension coefficient, and the molar extension coefficient is basically the measure of how strongly a chemical absorbs or attenuates light at a given wavelength. So to calculate the absorbance of any O, we need to know the molar extension coefficient as well as the concentration at which it's kept and the path length through which the light is passing. Intrinsic fluorescence of proteins is due to the amino acids which contain aromatic side chain. Then moving on to the basic amino acids which have a positively charged R groups. Basically side chain jo in amino acid mein hai, those are positively charged. That's why they are basic amino acids. We have lysine over here which contains alpha, beta, gamma, theta, delta carbons in the side chain along with an NH2 group which is positively charged, and arginine again similar, and it is called the guanidine side chain. We have, as we can see, this NH2 is positively charged and we have three NH groups. Histidine, the structure of histidine is this, which contain imidazole ring. The pKa is 6 for this ring, which is near neutral value. Histidine proteins, they are rich in lysine and arginine, and histamine is basically derived from histidine. Moving on to the amino acid with negatively charged R groups, basically acidic, we can say amino acid. There are two, which are aspartic acid and glutamic acid. So both of these, aspartic acid and glutamic acid, they can undergo phosphorylation. Polar uncharged side chain wale amino acids are serine and threonine. They are the OH containing amino acids. Basically, they have a hydroxyl group in them. They are responsible for the O-linked glycosylation that happens in the Golgi apparatus. Basically, glycoproteins का जो O-linked glycosylation होता है Golgi apparatus में, they are responsible. Serine and threonine are responsible for that, and also serine and threonine can also undergo phosphorylation. And tyrosine can also undergo phosphorylation. So basically we have 5 amino acids which can undergo phosphorylation, which include aspartic acid, glutamic acid, serine, threonine, and tyrosine. Moving on to the next sulfur containing amino acid is the cysteine, which is CCONH3H and then CH2 in combination with SH. When two cysteine amino acids, they combine, they form a disulfide bond within them, and this new amino acid that is created, it's called cysteine. That might happen sometime when a disulfide bond is formed between two cysteines. Asparagine is our next amino acid. This is the structure, CH2C double bond to NH2. We have an amide bond over here in asparagine. Glutamine is the same. C side chain consists of CH2, CH2, C double bond to NH2. The amino acids asparagine and glutamine, they are responsible for N-linked glycosylation that happens in the endoplasmic reticulum. So we know that serine and threonine are responsible for O-linked glycosylation in Golgi apparatus, while the amino acid asparagine and glutamine are responsible for N-linked glycosylation that happens in endoplasmic reticulum. So if we have an example, for example, the question they ask, phosphomimic mutants of protein can be generated by replacing what? So we have these two. Serine can be replaced by aspartic acid and threonine can be replaced by glutamic acid. As we all know that serine, threonine, aspartic acid, glutamic acid, tyrosine, these all can undergo phosphorylation. So any of the amino acid that can undergo phosphorylation, they can be interchanged and they can create a mimic mutant of that particular protein. Coming on to the amino acid that can act as acid or base, basically they have both positive and negative charge in them. They are known as Zooter ion or bipolar hybrid ion. So here we can see at this side, on the left side, we have a negative charge, which is the net charge is +1 because of this amine group. And on the right side, the net charge is -1 because of this carboxyl group. But when we see at the center, they have both the groups, the amine and the carboxyl group. So the net charge of this ion is becomes 0. This is called as Zooter ion or a bipolar or hybrid ion. This ion can act as acid as well as base. Isoelectric point, which is also known as Pi, it's the pH at which the net charge on a protein or an amino acid is 0. So how do we calculate the isoelectric point? We add both the pKa. We find the average of pKa1 and pKa2 values. And what's pKa? pKa is basically tendency of a group to lose protons or H positive ions, and it's important to mention that the pKa value is inversely proportional to the tendency of any group to lose or give up H positive ions. In layman's term, if we say pKa is the pH at which amino acid or protein loses H positive ions, and this inversely proportional situation suggests that higher the pKa value, lesser is the tendency of a group to give its H ion. So if the pH is equal to Pi, that is the isoelectric point, the charge on the group is 0. However, if the pH is greater than or less than the isoelectric point, the charge is present. Then we come on to ampholytes. Ampholytes are basically polycarboxy polyamino acids. Polycarboxy polyamino acids. They are used to create a pH range. So we'll have a strip of something, and throughout this strip there will be different polycarboxy polyamino acids will be present, which will be diverse in range. Matlab, from top to bottom there will be different pH in this strip. If pH is greater than Pi, protein has negative charge or is acidic and will move towards anode. If the pH is less than Pi, protein will have positive charge or is a basic amino acid and it will move towards cathode. So this polycarboxy polyamino acid, the ampholytes are used in isoelectric focusing. For example, it's used in 2D gel electrophoresis. So these are the pKa1 and pKa2 values for different amino acids which are given. So we have basic or positively charged amine side chains, and we have negatively charged side chain. pKar is that of side chain. So using these values, the isoelectric point of any amino acid is calculated. Okay, so that is it for right now.